To talk about streaking, I want to show you this plate from Lab 1. In Lab 1, we did an environmental survey where I went around and swabbed a few different surfaces around the college. If you take a look at this particular plate, this was the keyboard that I swabbed, and it has at least 4 different species of bacteria on it. There are these little yellow ones, this big creamy colony, some little tiny white colonies, and these opaque creamy colonies. So what I might want to do is study one of those species of bacteria individually. So what we do is something called a streak plate, and it looks something like this. So this is a streak plate for some sort of yellowish type of bacteria. So what is the purpose of streaking? The idea behind streaking is to basically take a culture and dilute it several times over a petri dish in order to produce individual colonies, and hopefully each of those individual colonies originated from a single bacterium so that you have a pure culture or a pure strain. This is particularly useful with a lot of environmental samples because you don't know necessarily whether your culture is pure or not. So if you want to study those organisms, it's important to isolate them so you have a pure strain so that you know exactly what you're studying. I'm going to show you now the process behind streaking, and then I'll put on a little demo that I did in the lab. To do a streak plate, what I'm going to do is get an agar plate, and I'm going to add an inoculation to part of that plate. So a little bit of bacterial culture. I'm going to streak it or smear it across that portion of the plate. Then what I'm going to do is sterilize my instrument. I'm going to turn my plate, and I'm going to take my sterile instrument, and I'm going to streak it through the original zone and spread the bacteria into a new zone of the plate. So hopefully at this stage what I'm doing is diluting that initial inoculum a little bit. I'm going to do that a third time, so rotate my plate, sterilize my instrument, smear my inoculant into a third portion of the plate. Then I'm going to do this one more time into the last portion of the plate. So rotate my plate, sterilize my instrument, and spread my inoculum into 4 quadrants of the plate. So here's a sample plate, and you can see what I've done is there's the 4 zones, and in zone 1 it's a relatively concentrated sample, and it gets a little more dilute in zone 2, a little more so in zone 3, and by the time you get to zone 4, if you've done this process right, what you've done is you've diluted enough that you're dragging individual bacterial cells into that 4th zone of the plate. Each of those individual bacterial cells will hopefully grow and then double again and eventually form their own colony. There are some isolated colonies, and each of those isolated colonies should be a distinct species or strain that have originated from the original cell. So what you can do is take those isolated colonies and now grow up a pure culture. So I'll demonstrate this in a moment, but what I want to do is talk about something called aseptic technique first. So what is aseptic technique? Sometimes it's called sterile technique. So this is a whole bunch of different types of practices and procedures that are basically trying to prevent contamination of your sample. So why are we doing this? 2 reasons really. We want to protect the environment. You're doing everything sterilely and carefully so that you're not bringing any bacteria home or spreading it around the lab. We don't want anybody to get sick. The other reason is we're actually protecting our experiment too. There's a lot of bacteria in your breath and on your skin, and you don't want to contaminate all of your samples. So what does aseptic technique include? It includes a bunch of different procedures, and depending on your lab and what you're trying to do, it could involve a bunch of different things. But generally what it involves is having all your equipment sterilized. So in this photograph here, you can see I have an inoculation loop, and it's actually getting sterilized by sticking it into a flame. A lot of glassware and plasticware can be sterilized using pressurized steam or ultraviolet light, and there's a whole bunch of different ways that things can be sterilized. This also involves reducing the time of cultures in the air, so you're not going to leave your culture with the lid wide open so that dust particles and whatnot can go into it. So you open your lid, you try to work quickly, put the lid back on. And lastly, of course, and this probably makes sense, you're going to be careful. So what I'm going to do now is I have a demo video where I'm going to do a streak plate. I'm also going to demonstrate a little bit of aseptic technique in that same video. Right now I'm going to demonstrate streaking using aseptic technique. So the first thing I need to do is turn on my Bunsen burner. There we go. Next thing I need to do is sterilize my inoculation loop, and I'll get there in a moment. I'm just going to talk a little bit about the Bunsen burner. The Bunsen burner obviously is a flame, and I'm going to assume that the area around the Bunsen burner is somewhat sterile because it's actually sucking in oxygen and air this entire time. So this whole area around the Bunsen burner is going to be kind of my sterile area. So as I mentioned, I'm going to sterilize my inoculation loop by putting it into the Bunsen burner flame until it glows orange. So that will incinerate any bacteria or organisms that are on the surface of the inoculation loop. The next thing I'm going to do is I'm going to get my culture, and what I want to do at this point is not put anything down on the bench. I have wiped and disinfected the bench, but it's not necessarily 100% sterile. So I want to keep everything in my hands and not put anything down on the bench. So the way I'm going to do that, I'm going to open this tube up, and I'm going to open it up and grab the tube lid with my baby finger, kind of like this. Keeping this near the flame, I'm going to flame the edge of the test tube just in case there's anything on there, any contamination on the test tube, and then I can stick my inoculation loop into the culture. So I'm going to grab a little bit of culture on the end of this inoculation loop, sort of like a bubble wand. I can return the cap to the test tube. I am now done with the test tube. I'm going to apply my culture to my agar plate. So flip over my agar plate. I'm going to spread the culture in one area, one quadrant of the agar plate. Now what I want to do is re-sterilize my inoculation loop. So notice I am putting the lid back on the plate. I want to minimize the amount of time that I have the plate open to the air just to avoid contamination. So re-sterilize my loop. I'm going to cool my loop off in an unused part of the plate. The plate itself is going to get rotated 1/4 of the way, and I'm going to spread my inoculation loop through where I have the old smear into a new part of the plate. So the whole idea here is to dilute the culture in order to get isolated colonies. Re-sterilize my loop, cool off the loop, turn my plate 90 degrees, and make a third streak. Do this one more time. There's a little bit of plate that still is unused. Re-sterilizing my loop. This is very important. It did have bacteria on it. I don't want to contaminate anything in the lab. Last step is to turn off my Bunsen burner, put a label on the plate, so this will be mixed culture. And I'm going to put this plate in the incubator and have it in there for a day or two at 37 degrees Celsius.